Hi everybody, welcome to our review session. We're going to talk about gene expression and the trip operon. Probably go over like RNA stuff on Friday, so I don't think we're going to get to that in this section, but we'll go over review stuff, and if you guys have any questions, please ask them. There's May and I's contact info if you have any questions later after the session. So first we want to talk about transcription regulation. So Laura talked about transcription factor mediated transcriptional regulation. So basically that sounds like a mouthful, but it's basically just how transcription is regulated. So why do we need transcription factors in the first place? Because RNA polymerase cannot bind to DNA on its own. It needs that transcription factor in order to bind. There's certain types of transcription factors. There's general and there's regulatory. General transcription factors aren't specific. They can really bind anywhere, and that is where we have our sigma factor for prokaryotes and the basal transcription factor for eukaryotes. You don't really need to worry about the basal transcription factor because in the trip operon, that's only in, we're only going to be talking about E. coli, which is bacteria or prokaryotes. So we're mostly going to focus on the sigma factor during this review session. Regulatory transcription factors are different from general because they are gene specific. There's two types, positive and negative. Positive is to enhance transcription when it's bound to DNA, and negative suppresses transcription when bound to DNA. It's not like good or bad, it's just kind of allows transcription to occur as positive and inhibits it if it's negative. And then RNA, so in order for the initiation of transcription to happen in prokaryotes, you need your RNA polymerase plus the sigma factor, because you need that sigma factor in order for RNA polymerase to bind to the DNA. Otherwise, it won't be able to bind and transcription won't occur. So there's this diagram here. It's actually bigger on the next slide just because I didn't think you could see it. But see right here, you have your start site, you have your promoter, RNA polymerase binds to the promoter, and this yellow dot is the sigma factor, so it needs to bind with the sigma factor in order to bind to the DNA. And then this is how RNA synthesis begins with transcription. You have your growing mRNA strand, the sigma factor can dissociate, and then it will rebuild, rebind if it needs to happen again. Does anyone have any questions on that so far?
No.
No? Okay.
Okay. So transcription factors are going to bind the DNA in a sequence-specific manner. As Rachel mentioned, in prokaryotes, these transcription factors are sigma factors. We're talking a lot about E. coli as an example. There's 7 different sigma factors, and sigma 70 is the one we're going to be focusing on. It's used in a normal growth phase, and it binds about 800 promoters in E. coli, and those different promoters will have a different affinity for sigma 70. And then sigma 70 must bind RNA polymerase before contacting promoter. So just focus more on the sigma 70 one. Other sigma factors will be expressed and used for different situations like stress responses, but we're really going to talk about sigma 70.
Oops, sorry. May, do you want me to do? I can do this one, or you want to?
Sure, yeah.
Okay. So like May said, the sigma 70 is the transcription factor we're going to focus on for E. coli. Also, like we said, it's necessary for the initiation of transcription in bacteria. Without it, RNA polymerase can't bind. Proteins that bind RNA polymerase to the promoter, that's what the sigma is. It is sequence specific. So as you can see, it binds at the -35 box and the -10 box in the promoter. The -10 box is the TATA box that we've talked about in class. I think -35 is the Pribnow box, but I don't think you need to really know the name for that. So initial unwinding takes place at the Tata box because there are more adenine and thymine base pairs, and since they only have 2 hydrogen bonds compared to cytosine and guanine, which have 3, they're easier to break. And sigma is sequence specific, but it does regulate a lot of genes and can bind to various sequences. So affinity changes depending on the sequence, and that's kind of when we talked a little bit about consensus sequences. But I know there's a good slide about it that Laura puts in her lecture notes if you hadn't, or her lecture slides if you hadn't seen them yet. But the consensus sequence is basically you have a bunch of sequences, and whichever nucleotide appears the most in each sequence, it's kind of just like it's just based on the frequency of the nucleotides that are in the consensus sequence. And then we have some diagrams here that usually help you visualize. You have your Tata box here, and the Pribnow box down here. And remember that negative just means downstream. Any questions? No?
I just have a quick question. I think I didn't know if she said this specifically, but I think because I asked her during lecture that the Tata box is in eukaryotes, but the -10 box is similar to it. It's just specific for prokaryotes. Or did she mean something else? I just want to clarify.
Like you mean the Tata box is only in eukaryotes and the -10 is different in prokaryotes?
Yeah, I think that's what she said.
I don't believe, I think the -10 is the Tata box. I'm not really sure.
I thought it was the Tata box too.
I think it's the same. The -10 box is the Tata box.
Okay, that's what I thought. But what I
Yeah.
asked in class, she made it seem as if like the Tata box was like a eukaryotic thing.
It's
And
in,
the box is different, but I thought it was like the
no,
same.
yeah, the
Sure.
-10 is the Tata box, and I'm pretty sure eukaryotes have it too. But just because the specific example is prokaryotes. But yeah, it is the same. The Tata box is the -10 box.
Okay, I just wanted to make sure. Thank you.
Yeah, no problem.
I think -10 is just where that sequence falls. Like
Yeah.
and like
Yeah.
in eukaryotes, it might not be. It could be at the same spot, but like it all depends on what
Yeah.
thing.
Yeah. She probably explained it that way, but I know sometimes it can be a little confusing. Okay.
So for gene regulation in prokaryotes, you're going to have operons, and operons are a set of genes transcribed under the control of a single promoter. So as you can see, there's a promoter, and then that has different genes within it, and then that can create like 3 different proteins. So the genes are usually transcribed together, like, and they function in the same processes. So it's not like A is going to be transcribed at a different time. B, it's usually just one long thing. So it's going to be polycistronic mRNA. So it's a single mRNA that codes for multiple proteins. And the transcription of operons is going to be controlled by different transcription factors, and those can be categorized as activators and repressors. So activators are going to increase transcription, repressors are going to prevent transcription. And activators and repressors can be turned on or off depending if they are bound to a certain molecule. And you'll see that in the trip operon.
Yeah, so just something to highlight specifically is the polycistronic mRNA. So if you see that word on an exam or a quiz, just remember that it's a single strand of mRNA that can code for multiple proteins. This is a good diagram of it.
I have a quick question.
Yeah.
Where does polycistronic mRNA come from? Like, does it just like pop up randomly when mRNA is being coded?
I think it's definitely in the genes. I don't know if I have the exact answer for you for that. But I know that only prokaryotes can have polycistronic mRNA. Eukaryotes can't. But
Okay.
it must just be in the genes when it's made.
Okay, that's fine. Thank you.
Yeah, sorry, I can't answer it better. I really don't know. But yeah, polycystronic's only in prokaryotes, not eukaryotes. Okay.
Yeah, I feel like it's just if the genes code for it,
Yeah.
then that's what will.
Okay. I kind of feel like that's a question that Laura would ask on the exam,
Where it comes from?
like where it comes from, or even something like do prokaryotes and eukaryotes both have it, or something like that.
Oh, yeah. Well, she could ask you that. I don't know if she would. I mean, when
don't
I
think
was in
she
her,
asked you that.
yeah. Just remember that polycistronic makes multiple proteins from one strand of mRNA, and that's only in prokaryotes, not eukaryotes.
Okay, thank you.
Yeah, no problem.
For like these topics in general, like we're focusing a lot on prokaryotes, so definitely know prokaryotes, and just kind of know what isn't categorized in prokaryotes. So know that there are basal transcription factors. You don't have to know in depth, know they exist, like that sort of thing. You want to know like what prokaryotes have that eukaryotes don't, but not like that in depth. She's not going to ask you to like ask where it comes from, pretty much.
Yeah.
Okay,
And
thank you.
yeah. Okay. So operons. An operon is basically just a combination of a set of genes, as well as the regulatory DNA sequences for that set of genes. So each operon has a promoter, and this is where the RNA polymerase will bind, and an operator, and that's where the repressors bind. The promoter is also where transcription begins, because that's where the RNA polymerase binds. Activators will bind to a sequence of DNA somewhere upstream of the promoter to recruit RNA polymerase. So as you can see in these two diagrams, this is where the activator is. It's upstream from the promoter region where the RNA polymerase will bind. So the activator recruits the RNA polymerase to bind at this the spot to start transcription. And then over in the second diagram, you have the operator where the repressor will bind, and it basically just blocks this RNA polymerase from binding, and no transcription occurs. And as you know, no transcription occurs, no mRNA is made. Operons can also be inducible or repressible. Inducible is when the operon is usually off, but can be turned on in the presence of a specific molecule. If you learned in your like bio 151 classes or 152, the lac operon is inducible. You don't need to do that for this class, but it's just an example. And then operons can also be repressible, where the operon is usually on, but can be turned off in the presence of a specific molecule. So the trip operon, which is what we're going to talk about, is repressible because we'll get into it. But when tryptophan's when it's in the presence of tryptophan, the operon's turned off. But we're going to get into more detail about that. But that means the operon is repressible.
Okay. So we have the trip operon, which is what we're going to focus on. So that's tryptophan, and tryptophan is an amino acid necessary for protein synthesis. So it's unique for bacterial cells in this situation because it kind of senses the levels of tryptophan and then is able to make tryptophan on its own. So tryptophan is kind of a regulator for itself in that sense. So we have two main scenarios: low levels of tryptophan and high levels of tryptophan. If you have low levels of tryptophan, the bacteria is going to need more tryptophan. So they're going to make proteins that will make tryptophan for themselves, and genes are going to be on. If there are high levels of tryptophan, you don't need to make tryptophan, so the genes are going to be turned off. So for this, control matters because if you already have tryptophan, you don't want to waste the cell's energy, bacteria's energy, to make it when you already have it. So in this sense, the trip operon is going to be repressible because you don't need everything if you already have tryptophan.
Does anyone have any questions so far on any of this?
I have a quick question.
Sure.
So I understand that when there's enough tryptophan in the cell, it can
Mhm.
bind to trip repressor to prevent RNA polymerase from binding to the operator,
Mhm.
thus essentially inhibiting transcription from happening.
Mhm.
However, I also understand that like when you're prohibiting this transcription, I know it's probably not the same thing as attenuation,
Mhm.
but when attenuation happens, RNA polymerase still binds and it's only half read because I know at some point ribosome is going to bind and then it's going to cause that hairpin loop
Mhm.
at like regions 3 and 4 because there's enough tryptophan in the cell.
Mhm.
How does the cell determine whether or not it's going to use trip repressor or it's going to use hairpin loops to inhibit the transcription of making more tryptophan?
Well, we're definitely gonna go into like everything that you just said, like all that, the mechanisms of attenuation and all that. Um, I think it's just there's like a threshold. It's just basically like if there's enough tryptophan, I guess it's like a threshold of what is enough and what isn't enough, if that makes sense. I like to think of it in terms of like, with an action potential, if you don't reach -55 or above, the action potential doesn't fire.
Mm-hmm.
Um, and then if you're below that, if you're above that, it will fire, and if you're below it, it won't. So that's how I like to think of it. I don't know if there's a better explanation out there, but it's usually the cell. It's really based on like how much it needs because tryptophan isn't that common of an amino acid to be floating around freely. So when it is, I think the cell kind of is sensitive to that. But when there's absolutely no tryptophan, like very, very low, that's when the trip genes are on and you're going to make tryptophan, you're going to do the full transcription. And then there's that like threshold of what's too high for in terms of levels of tryptophan.
Okay.
Does
Because
that make sense?
yeah, I think
Okay.
like in her topic notes, she
Mm-hmm.
mentions that like really high levels of tryptophan, because you're gonna have more of it in the cell, then
Mm-hmm.
repressor is gonna cause that blocking of transcription. But if you just have like medium levels,
Mm-hmm.
then you're gonna have that hairpin loop because like
Yeah.
there's not an abundance of it for repressor to go and bind all the time. So like
Mm-hmm.
RNA polymerase may bind at times, but because like the ribosome and everything can sense the levels, it's only gonna um, stop over region 2, causing the hairpin loop. So it's like kind of a backup to repressor when there's not high enough levels of tryptophan in the cell.
Yeah, we're gonna go like through the attenuation process and that like medium level situation when it's not high, it's not low.
Okay.
So like maybe we'll go over like answer like more questions about that during
Oh,
when we go to that.
When you
cool.
said
Thank you.
medium level, um, think of that as kind of probably what I think that's what happens more often in the cell. So in the bacteria, so when you have really high levels of tryptophan, that's just saying like it's the levels are extremely high. It's not even it's above medium. So it's like so high you don't need anything, and that's when the repressor would come in. But if it's medium levels, then it's kind of like as Rachel said, it's as what the cell needs. So you're gonna have that partial. You're gonna have the attenuation more because you still need tryptophan, but you don't need as much as you would if the levels were really low.
Okay. That makes more sense. Thank you.
Okay. Yeah, so this is just a diagram of the trip operon structure. This is the trip R, which codes for the repressor. And then you have your P region, your O region. She talked about that in class today. The RNA polymerase will bind at the promoter, and the operator is where the repressor binds. And then this region right here, that's kind of, can you guys see my mouse? I don't know if I'm like pointing and you're not seeing where I'm pointing at all. Can you like see where I'm
Yeah,
pointing?
we can see
Yeah, I
it.
see
Okay.
it.
Okay. I was like, they probably can't see. Okay. So this region where it's like brought out, this is the leading strand, and this is where of the mRNA, this is where attenuation will happen. This is a simplified version. We're going to go into more detail about it. And then these are your genes that code for the enzymes that will make tryptophan. And then just, I think we talk about this in the next slide, but tryptophan is a co-repressor because it binds the repressor molecule and changes its conformation, so it has a higher affinity for the operator. But we talk about that in the next slide.
Yeah, so this is your first situation where you're going to have strictly like high levels of tryptophan. So the repressor is going to be bound to the operator, and then tryptophan is also going to be bound. So there's no initiation of transcription or translation whatsoever. That's basically that one.
Yeah, so basically tryptophan just binds to this repressor and changes its conformation, so it has a higher affinity for the operator, so it's like harder to dissociate, so it even prevents RNA polymerase even more from binding to the promoter. So this is kind of what we were talking about with the attenuator region. So this region right here, this diagram is back like 2 slides, is this leading strand right here after the operator. So you have your hairpins and your stem loop. Regions 1 and 2 are complementary, 2 and 3 are complementary, and 3 and 4 are complementary. So attenuation determines how long the mRNA transcript will be. It happens after transcription begins. So it affects how things get completed, and it's just a more fine-tuned level of regulation beyond just transcriptional regulation. It does so, like I just said, it does not affect transcription initiation. And basically, I think, yeah. Basically, when there's enough tryptophan around, then a short transcript will be made, and the transcription stops before the enzymes like are coded for. And then if there is not enough tryptophan around, then the full mRNA transcript will be transcribed. So basically, why do prokaryotes use attenuation? So they transcribe and translate at the same time because there's no nucleus, so it just happens in the same time. There's like going into a lot of detail about attenuation. Is this something that you guys get confused about? Because I know when I was in her class, it was tricky for me to kind of get a grasp on.
Yeah, it actually is kind of
Yeah.
confusing.
Yeah, so trying to think of the best way to explain this. So you have this leader peptide coding region, and then this is your region 1, your region 2, your region 3, and your region 4. So basically, the hairpin for 1 and 2 forms because the RNA is complementary. Then the RNA polymerase pauses. So the RNA polymerase, since it's happening at the same time, you have the RNA polymerase going first, and then you have the ribosome following it. So you have your transcribing, and then the ribosome's coming along after to translate it into the proteins. So after 1 and 2 form, you see how it says pause. The RNA polymerase pauses to give the ribosome time to bind. The most important thing, or one of the most important things about this, is that the RNA polymerase and the ribosome need to coordinate with each other in order for this process to work, kind of like a tag team. So this leader region is where the ribosome will bind at the start codon, and it will start translating as the RNA polymerase is transcribing. So another big thing to know is that ribosomes need to translate along single-stranded RNA. So when it comes along this 1-2 hairpin, it breaks it or it makes it back into single-stranded RNA because it can't translate double-stranded. So then the RNA polymerase will keep transcribing. The movement of the ribosome and the RNA polymerase are coordinated, which is crucial to work. And then when the ribosome reaches the tryptophan codons, this is when the RNA polymerase is working on transcribing region 3 right here. So it's after it breaks 1 and 2, and now it's over here. So this is when it varies. So if there's enough TRPT RNA present, the ribosome will translate through position 54. You don't have to know these specific details, but basically what happens if there is enough TRPT present, then a shorter transcript will be made because the ribosome finishes at the stop codon and pauses and then it rests on region 2. So then the ribosome makes region 3, then region 4, and the formation of the hairpin between 3 and 4 causes the end of the mRNA to I don't want to say, it jerks out of the active site, if that makes sense. And then transcription ends. So when you have enough TRPT, this 3 and 4 hairpin is created, and a shorter transcript is your end result. If the TRPT levels are low, then the RNA, so basically everything that I said before continues to happen. But attenuation does not occur in this situation if the TRPT levels are low enough. And then 2 and 3 will form a hairpin. So basically 1 and 2 will always form at the beginning for whether or not there's not enough or enough tryptophan tRNA available. Then if there's enough tryptophan available, then 3 and 4 will be formed, and you'll get a shorter transcript, and there'll be termination of transcription. If 2 and 3 are formed from when you have not enough tryptophan, then you will continue transcription. You will make the whole mRNA transcript, and you will make those enzymes that will make more tryptophan. I know that was a mouthful. I'm so sorry, but does anyone have any questions?
Yeah, so you said that 1 and 2 will always be made regardless of the amount of tryptophan, right? And let's see, 2 and 3 would be made if it has a high amount?
If it has a low amount.
Okay.
And 3 and 4 will be made if it has a higher amount.
Okay, thank you.
Yep.
So just repeating 1 and 2, they'll form a hairpin loop every time, whether it's going through attenuation or it's just transcribing.
Yep.
But they'll essentially form that hairpin loop between 1 and 2. Ribosome will come in, kind of just break it apart, and then depending on what it needs, whether it needs to have that early termination or not, it'll form either the 2-3 hairpin, which is the non-attenuation, or it'll reform the 1, 2, and 3, 4 for early termination.
Correct.
Okay.
Yes. So yeah, that's sorry. So 1 and 2 will always form. Then that ribosome will break them apart because it needs that single-stranded RNA. And then that's when the cell determines whether or not it needs more tryptophan. If it does, then you'll have 2 and 3 form, which will continue transcription and translation. You'll get those enzymes that will code for more tryptophan. If you have the cells oh, this is way too much tryptophan, I don't need this much, 3 and 4 will form, and you will stop transcription. And that stopping of transcription early with that 3 and 4 hairpin loop is called attenuation. So when you have 2 and 3 together where you're continuing transcription and translation, that is not attenuation. Does anyone have any other questions? I know it was a lot, but that's the basics of it that you really need to know with those different hairpin loops. And remember that attenuation happens after transcription's already been initiated. So it doesn't affect whether or not transcription has started or will start. It already is starting.
I have another clarification question.
Yeah, what's up?
So I know that RNA polymerase is going to be transcribing what's on the mRNA, but when ribosome binds, is that the part of this process that's translating? Because
yeah,
I know it's doing both.
Yeah, it's doing both at the same time. So basically you have this RNA polymerase going along this, this is DNA, and the ribosome's translating that mRNA that comes out of it, right? Yes. This is RNA. I'm sorry, this is mRNA. This is being transcribed by the RNA polymerase, and the ribosome is coming along behind it. So the ribosome is translating this. This is mRNA right here.
Okay, so the RNA is the transcribing, the ribosome is the translating.
Yes. Yep.
Sorry, I just have one last clarification. Sorry.
Yeah, you're fine. I know it's a lot.
So attenuation you said happens after 3 and 4, not 2 and 3, and this happens after transcription, not translation.
Yes. So attenuation is the termination of transcription in 3 and 4.
Okay.
And then 2 and 3 is just the regular, the regular process. Like you have your transcription and then translation happens. But 3 and 4 is that shorter transcript where the enzymes aren't translated for because the genes aren't, like, you're not made, you don't get to make those, the proteins for the enzymes because translation doesn't happen.
Okay, thank you. Yeah, actually, the thing was that I actually took a screenshot of the slides. I'm typing out the notes next to it while you're saying it.
Yeah, no, yeah, you're good. Does that make sense? So like the 3 and 4 is attenuation when you have that shorter transcript. And because if you think of it, like this is that leading strand, so this is right after the operator. So the genes that code for the enzymes are further down. And if you're stopping here, you're not going to make those genes that code for those enzymes to make tryptophan. So that's why when 3 and 4 are formed and you get this termination, you get this attenuation, it happens when you have too much tryptophan. So the cell started and was like, oh, like I need more tryptophan. And then it realized, wait, just kidding, I have too much. So it stops. You don't make those enzymes that are further along in this sequence, and that's attenuation. But with 2 and 3, you will still end up making those enzymes.
Yeah, thank you. This makes a lot more sense now.
Okay. Good, I'm glad. I think May also included a YouTube link in the notes for this. I watched it too. It is actually pretty helpful.
I can put it in the chat right now.
Yeah,
there we go. Oops. That's kind of like where the pictures came from, but it goes, I think it's detailed. It's nice.
Yeah, it was really nice. Any other questions? I know it's confusing, but try to simplify it as much as we can. Okay, cool.
So hopefully the next few slides will help you visualize it a bit more. So this second situation is when you have the low levels of tryptophan and you want to make more. So your repressor isn't going to be bound to tryptophan and is not bound to the operator. And for this one, since you want more tryptophan, attenuation isn't going to occur. You're going to have your full transcription and translation, so that means all 4 regions of the attenuator sequence. So your first step, you're going to have your 1-2 hairpin form, and that's going to let the ribosome pause and it's going to let it catch up to the RNA polymerase. When this happens, the ribosome is going to get stuck on the trip codons in region 1. Because it's stuck on the trip codons in region 1, RNA polymerase isn't affected by anything and it can still transcribe. So it's going to transcribe regions 2 and 3. And after that happens, regions 2 and 3 can then hairpin with each other because they aren't occupied, they aren't affected by region 1. Sorry, they aren't affected by region 1. So because that happens, 2 and 3 can hairpin. And that 2 and 3 hairpin is your anti-termination signal. That signals that transcription is going to go the full length and you're not going to end it early. So that is when you're going to get your full mRNA transcript. Does that sort of make sense?
Yeah, so this only goes up to hairpin 2 and 3, right? 3 and 4 is not included.
3 and 4 aren't going to hairpin, but you're still going to transcribe the whole thing. So what happens is regions 2 and 3 hairpin, and that signals that transcription is going to go the full length 1, 2, 3, and 4. And so pretty much since 2 and 3 hairpin, 2 is occupied and 3 is occupied. So because 3 is bound to 2, 3 can't be bound to 4.
Okay.
So that means there's no possibility of a 3,4 hairpin.
Okay, I see. Thank you.
Is there any other questions on this? These diagrams definitely make it clearer, as you can kind of see, like this RNA polymerase is transcribing while the ribosome translates the mRNA that comes out of it, right? So any other questions? No. May, did you want to say anything else about this slide?
No, I think that was it.
Okay.
Yeah.
So this is more about the attenuation, where you have initially low levels and then it goes to high. So this is kind of like the cells like, I need more tryptophan, but then it's like, oh, nope, just kidding, I don't. So attenuation, like we said before, transcription, translation is you already started transcription, but translation gets stopped. So this transcription's already happening. And then, so the 1 and 2 hairpin form, like they always do, the ribosome catches up, breaks it apart because it needs that single-stranded RNA. Once the ribosome reaches the trip codons, there's enough tryptophan to make tRNA, and the ribosome does not become stuck on region 1. It now gets stuck on this stop codon over here. So it reaches the stop codon of region 2 and is now stuck here. By blocking region 2, regions 3 and 4 hairpin, and this is the termination signal for attenuation. So this, see down here, this is where you have 3 and 4 that make that hairpin, which causes the termination. This results in the short mRNA transcript. RNA polymerase dissociates from the DNA mRNA, and you stop both transcription and translation. So this is that termination signal when you realize you have too much tryptophan. You started low, but now you're too high, and you don't need more.
Yeah, so this was
Any
like
questions?
the medium levels we were talking about.
Yeah.
So this is just to recap. You have your situation 1 where you have high levels of tryptophan and you don't want to make any more. So your repressor is going to be bound to tryptophan and they're going to be bound to the operator, and there's no initiation of transcription or translation. Situation 2, you have your low levels of tryptophan and you want to make more. So the repressor and tryptophan are not going to be bound to the operator. You have your 2-3 hairpin loop that will signal that there is no attenuation that is going to happen. So you're going to result in your full transcript. And then situation 3, it's your medium or low to high levels of tryptophan. Transcription is started and your 3-4 hairpin form to signal attenuation, and it's going to stop transcription early, and you're going to result in a shorter mRNA transcript.
Any questions? That was the last like main slide we had for you guys. So if you have any
Could you repeat the second situation again? I'm kind of confused.
So that was when we had the low levels of tryptophan, and if the bacteria has low levels of tryptophan, it's going to want to make more tryptophan and increase the levels of tryptophan. And because of this, you're not going to repress the operator with a repressor or tryptophan because you want transcription to occur. And you're going to have your regions 2 and 3 hairpin to signal anti-termination. So that means you want no attenuation to occur and you want the full transcript at the end. So that means you're going to have your full transcription and full translation processes.
Do you mind go back to the slides that describe this situation?
This one?
Yeah.
So yeah, so this is your low levels of trip. Your repressor's not bound because you need more tryptophan. No attenuation happens because you want that full transcription and translation because you want those enzymes that are further along in this operon to be coded for so that they can be transcribed into this mRNA and then translated into the enzymes that they need to be so they can make tryptophan. So basically the 1 and the 2 hairpin form, whether or not you have low levels of tryptophan or you initially start low and then go to high. The only time the hairpin doesn't form is in this first situation when you have high levels of tryptophan because transcription just doesn't happen. So basically, this hairpin forms, the ribosome and the RNA polymerase work together. The RNA polymerase is not affected, so it does transcribe regions 2 and 3, which then form the hairpin loop. Because when 2 and 3 are together, then 3 and 4 can't make that termination signal. So this 2 and 3 hairpin is the anti-termination signal, so it results in the full mRNA transcript. Because you want that full transcript to be made because you want tryptophan to be made from the enzymes that this operon codes for.
So in this situation, the RNA polymerase will keep transcribe, and then ribosome will like break the bond between region 2 and 3, and then keep translate.
Yeah, because
Okay.
it's going to want to translate the whole code because it needs to make those proteins out of those for those enzymes to form.
And then if region 3 and 4 form the hairpin, ribosome will not break it.
No, because this ends in termination.
Okay. Okay. So only 3 and 4 from the hairpins, ribosome will meet the terminal.
Yeah, it's more about that termination signal for the RNA polymerase to dissociate. So that transcription stops and translation stops because there's nothing for it to translate since RNA polymerase isn't transcribing anymore from this initial DNA signal.
Could you repeat that?
Yeah. So, sorry, a helicopter is flying over my house. So the termination signal is for the RNA polymerase. Hold on. Wait a second.
Sorry, I think it's flying over my house too.
Yeah. Can you guys hear me now? Sorry. So the termination signal is from the 3 and the 4 hairpin down here, which causes the RNA polymerase to dissociate from the DNA and the mRNA. So because it dissociates, it's no longer transcribing this DNA from here. Since it's not transcribing anything, there's nothing for the ribosome to translate, which results in this shorter mRNA transcript. It kind of just stops right here.
Okay.
And there's really nothing for the ribosome to translate.
Oh, so is the RNA polymerase stop first and then ribosome stop? Okay, thank you.
You're welcome.
I have a quick question about one of the problem set questions that kind of relates to this stuff. So it's problem set question number 2, but it mentions that if the trip codons were mutated into stop codons, how this would affect the regulation of the operon when there's medium amounts of tryptophan. And so what confuses me about this is the fact that I understand that when there's low levels of tryptophan, ribosome's gonna stop over those tryptophan codons and wait for a tryptophan tRNA to bind so it can make more before moving along. But if it stops at that same spot, but instead of the tryptophan codons, they're stop codons, how is that affecting the cell? Because if I'm thinking about it in like any normal way, when a ribosome stops at a stop codon, it just stops, and then I think at some point it like begins again when RNA polymerase is ready. I'm just confused how that's going to correlate with tryptophan if the tryptophan codons have now been replaced with stop codons.
So if the tryptophan codons are replaced with stop codons, I mean, you're not gonna get any tryptophan because you're stopped. Did you say this is from the problem set?
Yeah, it's from problem set this week. It's
Oh,
question
okay.
number 2A. I was at like a help session earlier, but like we were talking about it for like a while, and like we were
Yeah.
started getting really confused. So the TA just said to bring it up during the session.
Isn't that question 1B?
It's 2B, or maybe it's 1B. Whichever one it is. I like renumbered mine, so that's probably why it's.
Oh, okay. Okay,
Yeah.
no, okay.
Sorry.
So, no, you're totally fine. Presence of
It's
low.
like in the presence of low medium concentrations of tryptophan.
Yeah.
That problem.
Okay. Primarily short.
Yeah, so having all those stop codons would make the ribosome stop a lot.
Like, would ribosome ever move forward after that? That's the thing. Like, I actually really don't know. Like, I know ribosome has to stop at a stop codon, but like, I don't even know if it ever moves, or does it unbind? You know, I'm not really sure what it does after that. And if that's where the tryptophans are, like, it's obviously not going to be able to make tryptophan when it needs it, or it won't make tryptophan tRNA. But I know that also correlates to levels of tryptophan in the cell. This is where I start to get a little confused.
Mm-hmm. So, if you change the trip codons to stop codons, the ribosomes will stall there every time.
Mm-hmm.
Because it does, in the question, you know, how she says, like, the ribosomes typically pause or stall at stop codons. So if you have a lot of them, it's going to stop a lot, which means it's going to translate very slowly. So eventually, so if the ribosome is not translating enough, I'm trying to phrase this question better. Okay, so if the ribosome is not translating a lot, what is so if it's not translating quick enough, what environment will the cell eventually be in? Like in terms of tryptophan concentration?
It'll be in low concentrations.
Right. So when you have this stop codon that's stopping and it's translating very slowly, even though it's trying to do the whole transcript, it's going to be very slow because you're not going to be making those enzymes quick enough to make more tryptophan. So eventually the cell will think it's in a very low tryptophan deprived state. So if the cell thinks it's in a low tryptophan deprived state because it's not translating quick enough because you have all these stop codons, what type of transcript will be made? Like, will attenuation happen?
No, attenuation won't happen.
Right. So the whole mRNA transcript will be made. Does that make sense?
Yeah.
Okay.
So the whole mRNA will be transcribed.
Mm-hmm.
And when it's transcribed, is it making more tryptophan? Like, what is the mRNA that the RNA polymerase is transcribing? Like, what is it making?
So the RNA polymerase is reading this DNA from the operon and creating this mRNA transcript. So that's your transcription. Then the ribosome comes in and translates the transcript into proteins. You know how you'll have, like, it'll make each amino acid for the protein, but it's not making tryptophan, it's making the enzymes that will make tryptophan. Does that make sense?
Okay, so the mRNA
Mm-hmm.
will make, like, when translated, when mRNA is translated for this, it's going to make enzymes that make tryptophan?
Yes.
Okay. So when, like, with the stop codons, like, it's stopping at every stop codon.
Yes.
So the ribosome will stop at every stop codon it runs into.
Mm-hmm.
But it's still translating. How long does it stay at the stop codon? And, like, when does it move or continue translating?
Honestly, don't know how long it stops at the stop codon. I don't think that's something she would expect you to know either.
Mhm.
It kind of just gets stuck for, I mean, I really don't know how long, but it's enough to make the cell think it's in a tryptophan starved state.
Okay. Is another reason why it's in a tryptophan starved state is because it no longer has that like tryptophan tRNA like sensing sequence.
I don't know if that's it. I think it's just the fact that the ribosome is just translating very slowly.
Okay. Yeah, that was the part that I was confused because I'm like, well, if they're getting rid of the tryptophan sensing, then how's it going to be able to sense anything about the levels in the cell?
Yeah.
That was the part that was just like really
Yeah.
boggling my mind. But I think that clears things up. So it's just going to stop at the stop codons, and because of how slow the translation's happening, the cell's going to think that it's in a tryptophan low state, even if it's not. And so therefore RNA polymerase will continue to transcribe the entire mRNA sequence. And that's it.
Yeah.
So you'll end up with more transcribed mRNA as a result.
Yeah, that's what I would say, yeah.
Okay.
I don't know if Mayo has anything to add, but that's basically.
Yeah, that's basically it. I think she mostly just wanted you to focus probably on the frequency of the ribosome stopping and just realize how that mimics the tryptophan low levels.
Okay. All right.
Actually, while we're on the topic, can I actually ask a quick question about 1A? I already answered it, but I was a little iffy about my answer because it wasn't really clear during the lecture today.
Sure.
So for A1, it says that if the leader sequence region is deleted, basically just asking like what would happen. And so when I asked the question, the answer I got was pretty much if the leader sequence is deleted and still have the promoter and genes, then the cell will still be able to transcribe because the leader sequence is a secondary regulation mechanism, not the primary. Was that right?
So I'm just reading the question real quick, sorry. So what did you say is your answer again? Sorry, I was reading the question.
That's okay. If the leader sequence is deleted and still have the promoter and genes, then the cell will still be able to translate because the leader sequence is a secondary regulation mechanism, not the primary.
So in this specific example, if the leader sequence is deleted, then you still have that regulation of the TRP repressor, which would be now your only regulating component. But I would say the tryptophan would still be synthesized. I would agree with what you said. So, yeah. I'm trying to think of how to word this. May, what do you think?
I would say think about what is in the leader sequence and what mechanism would be taken out if the leader's sequence region was deleted. That has to do with tryptophan levels, how that affects tryptophan levels.
Okay,
I don't
and
know if that
so
makes sense at all, but
no, it did. It's basically just me having to go back and just look at that section of the lecture to see like how I can like make it a bit more of a coherent argument.
yeah, I'm looking at the question right now. A keyword is in the second sentence of that. I think you'd want to incorporate.
Yeah.
I would incorporate how the lack of a certain mechanism would affect.
Oh, I see it. Yeah. So then if that's the case, then for the second part of part A would be the mutation wouldn't be bad because it will still it will still be synthesized.
I wouldn't think of things in terms of good or bad. Well, actually, she said bad. Never mind. Never mind. Never mind. I'm like looking at it. I think, okay, so yeah, you're right for what you said. But take that a step further and think of what other process or how making so much tryptophan, how that would affect the cell. It's in our, like when we send out the slides. It's in our slides. We went over this. So I don't know.
Okay, I
Rachel, do you
see.
have anything else for
It's hard to like describe what she's like looking for without giving you the exact answer. But yeah, I would definitely look at that second sentence. And definitely think about like levels of tryptophan in terms of what is synthesized.
Okay, yeah, I was just a little iffy about the second half of that because
Yeah.
there's really no such thing as good or bad in this scenario. It's just how you look
Right.
at it.
Right. But yes, she did say that.
Yeah, she said
I
bad,
think if
and
you-
yet she's like, don't say it.
Yeah.
Think of like the Goldilocks hypothesis, sort of.
Actually, yeah, that's a good point.
I would just explain both because, like, in a way, you could kind of argue that it could be good or bad for the cell. You know, she's never gonna like dock you for more information as long as you explain the reasoning. So if you're not sure if it's like good or bad, or you see reasons as to why it's both, I would put both down. Because like you're right, like the levels at which it's going to produce, like it's not necessarily going to kill the cell, but it is going to affect how the cell regulates the amount that's in there because it doesn't have that leader sequence. So without that like leader sequence, it's not going to be able to sense how much tRNA is in the cell. So you'll have an abundance of it. So you could argue that in and of itself is bad because it was evolutionarily put in there to prevent something like that from happening. However, it's not detrimental because the cell isn't going to operate because there's still tryptophan available. Because if no tryptophan was available, the cell would die. So you could argue it like that by saying that, you know, it's not great, but it's not going to kill it.
Yeah.
I hope that helps because that's kind of how I answered the question.
Oh no, that actually definitely helps because I actually had no idea how to answer it because my mind was just battling back and forth with two possibilities.
Yeah, so I would just put both down because more often than not, she's not going to penalize you for more detail, especially if you explain why.
All right, I'll do that.
Yeah, so think of like what the cell, like you're looking at a mutant that is definitely producing tryptophan, right?
Yeah.
And then go from there. Think of all of the things that a cell has to prioritize to function and all the processes it has to do. So think of how producing so much tryptophan could affect that.
Okay, definitely, I'll do that. Thank you guys so much. Seriously, that
Yeah, you're
really helps.
welcome.
Thank you both. Hope you guys have a great night.
Oh, thank you. You too. Is it just us? It is. Okay.
I think so.
That made as much sense as it would to me if I were, I think, a student. But
Yeah, I was just like, I don't know.
no, I know. Well, okay. I think that went well.
We can stop the recording.
Oh yeah. So.